Welcome to section 1 of reproductive embryology. In this section, we'll be discussing early fetal development. Let's get started. This is an overview figure of early fetal development. We'll walk through each part one step at a time, so don't let it overwhelm you. On the left, we can see a truncated image of the uterus and ovary. Once the oocyte that you can see right here is released and comes in contact with sperm, it undergoes fertilization and is known as a zygote. As you can see, this is considered day 0. By day 2, the zygote has divided into 2 cells, and this is known as a blastomere. By approximately day 4, it has undergone multiple divisions and is known as a morula, which is a ball of about 10 to 30 cells. By day 5, a blastocyst forms, which is comprised of 2 distinct cell lines and a fluid-filled cavity in the center. This occurs as the cells of the morula secrete fluid and displace some of the cells to one side, leaving a fluid-filled area in the center. By about day 7, the blastocyst implants on the uterine wall, which is about the time that HCG secretion begins. Right here, we can see some of these structures better. So the cells surrounding the blastocyst are collectively referred to as the outer cell mass. And as you can see, this is also known as the trophoblast. The cells within the blastocyst are collectively known as the inner cell mass. You can also see some trophoblastic cells on the outside, which we've shown in green, and notice that these are beginning to invaginate into the uterine wall. If we zoom up on this blastocyst a bit later in time and we look at the developing structures from a cross-sectional view, we get this image over here. So by week 2, a bilaminar disc has formed. And this just means that there are 2 layers of cells stacked on top of one another, which you can see right here. So you can see the blue layer on top and the brown layer on the bottom. In reality, the bilaminar disc is a 3-dimensional structure and would look something like this. So imagine that we have 2 hamburger buns stacked on top of each other, and we're looking at them from the side. If we take a cut out of the hamburger like this, and look at the cut head-on like this, we end up seeing a cross-section of the bilaminar disc, which was what we showed on the last slide. So if we return to this view, notice that we're looking at a cross-section of the bilaminar disc, which is right here. Okay, before we go any further, notice that this mass of cells has successfully implanted within the uterine wall, so we can see the uterine epithelium right here. We can also see that the outer cell mass has divided into 2 distinct cell types, the syncytial trophoblast and the cytotrophoblast. The inner cell mass has also divided into the epiblast and the hypoblast. Again, these 2 cell lines form 2 layers, which is why this is referred to as a bilaminar disc. We can also see the beginnings of the yolk sac and the amniotic cavity. Sometimes it can be confusing to learn all these structures out of context, so I think it can be helpful to think of what these ultimately become. If you look ahead a bit, you can see that the epiblast becomes the developing embryo. So here we can see that the epiblast gives rise to these pink cells over here, and then that this color is maintained right here, and ultimately becomes the fetus. We can also see that the hypoblast in brown becomes the yolk sac right here and the umbilical cord, which we can see right here. The syncytial trophoblast and the cytotrophoblast become the placenta and the chorion. You should also know that in the early stages of fetal development, the syncytial trophoblastic cells secrete HCG. Additionally, these cells lack MHC1 expression, which is important because it means that the maternal immune system is less likely to create an immune response to the developing placenta. So again, if you're confused about what each structure becomes, just look at the corresponding color towards the end, and you can see what the early structure ultimately becomes in the more formed, developed fetus. So remember, by week 2, we have 2 layers, which is also known as the bilaminar disc. And this is composed of the epiblast and the hypoblast. All right, with this in mind, let's move on to discuss gastrulation, which occurs during week 3. If we zoom up on the bilaminar disc right here, during week 3, we can see week 3 right here, we get this image right here. As you can see, the epiblast begins to form a single ridge with a central area of invagination, and this is known as the primitive streak. So again, if we go back to this hamburger idea, and we're looking at the bilaminar disc from the side again, we should see a midline invagination begin to form like this. And this is known as the primitive streak. Hopefully with this drawing, the name makes a bit more sense. The primitive streak is across the length of the bilaminar disc. So I guess you could say it's a streak. And just like before, if we take a cut out of the hamburger like this, and we look at the cut head on like this, we end up seeing only a small segment of the primitive streak. So again, this streak is right here, which is looking at it from a cross-sectional view. And we can see that the cells from the epiblast begin to migrate through this area in between the epiblast and hypoblast and form the three essential layers of the developing embryo: the endoderm, mesoderm, and ectoderm. Endoderm formation occurs first and is the bottom layer. Then the mesoderm, which is the middle layer, forms. And finally, the ectoderm, which is the top layer. Sometimes students get confused and think that the hypoblast becomes the endoderm, but this is not the case. The epiblast forms all three layers of the developing embryo, and the hypoblast gets displaced downward. As I mentioned earlier, the hypoblast becomes the yolk sac and umbilical cord. So in summary, during week 3, 3 layers are formed. This occurs as the epiblast invaginates downward, which results in the formation of a primitive streak. The cells from the epiblast begin to migrate through this area, which is called gastrulation, and this results in the formation of the endoderm, mesoderm, and ectoderm. Around week 3 to 4, the amniotic sac enlarges, the heart begins to develop, and the neural tube forms, which is the embryonic precursor to the brain and spinal cord, and also the limbs begin to form little buds. If we look at the image, we can see that the epiblast has become the developing embryo, and we can see 3 distinct layers here, although they're not labeled. So it's a bit hard to see, but the top layer right here is the endoderm, and then directly below that, we can see another layer, and this is the mesoderm, and then this bottom layer right here is the endoderm. We can also see the neural tube, which is that circular structure in the middle right here. Notice that the amniotic cavity has completely surrounded the embryo, and the hypoblast has become the yolk sac, which, as I said before, is displaced downward. We can also still see the syncytotrophoblast and the cytotrophoblast. So in summary, during weeks 3 through 4, the amniotic sac enlarges, the heart begins to develop, neural tube formation occurs, and limb buds begin to form. You should also know that because there is so much going on during this time, and really from about weeks 3 to 8, the fetus is very susceptible to teratogens. Around week 5, the umbilical cord forms and many of the vital organs continue to develop. We can see that the yolk sac has become the umbilical cord right here. This is a bit of an oversimplification because other structures are involved in the formation of the umbilical cord, such as the allantois. But for simplicity, just think of the hypoblast and the yolk sac becoming the umbilical cord. We can see that the syncytotrophoblast has become the chorion and the outer layer of the placenta shown in green right here. The cytotrophoblast has become the inner layer of the placenta shown in this kind of pinkish color right here. From here, organogenesis continues, and around week 12, most of the organs are developed. Now, if we change the perspective a bit, and instead of looking at the fetus from a cross-sectional cut, we look at it from a coronal cut, we can see something that is very easy to identify. We have the developing fetus inside the uterus right here, and we can see the chorion, amnion, amniotic cavity, umbilical cord, and the placenta directly adjacent to the uterine wall right here. So I talked about the syncytotrophoblast and the cytotrophoblast with regards to this image, but notice that if we look at the beginning again, the cytotrophoblast is pink right here, and the syncytotrophoblast is green right here. This continues all throughout the image. So if we look at the final image, we can see that the syncytotrophoblast has become the chorion, which is this outer green layer, as well as the outer layer of the chorionic villi right here. Now, the cytotrophoblast is pink, which we again can see right here. And if we follow that to the end of the image, we can see that this now is the inner layer of the chorionic villi right here. Next, let's go over a few more major milestones between weeks 5 and 12. By week 6, fetal cardiac activity is visible by transvaginal ultrasound. By week 8, fetal movement starts, and by week 10, the genitalia now have male or female characteristics. And on the right, we can see an ultrasound of a 10-week-old fetus. The fetus is right here. All right, now that we've covered the image, let's review with a question. A 24-year-old pregnant woman comes to the clinic due to dysuria and urgency that began yesterday. She states that she has leftover pills of TMP-SMX, or Bactrim, from a previous UTI infection and is wondering if she can take this medication. If she takes this medication, at what stage in development would her baby be most susceptible to the adverse effects of Bactrim? A, 1 to 4 days, B, 6 to 10 days, C, 2 weeks, D, 4 to 6 weeks, or E, 10 to 12 weeks. Hopefully from the question stem you notice that she was thinking about taking TMP-SMX, which has teratogenic effects. Fetuses are most susceptible to teratogens during the embryonic period of 3 to 8 weeks because this is when important organs are forming. So the correct answer is D, 4 to 6 weeks. 1 to 4 days is incorrect because this is when the egg is fertilized and divides to become the morula. 6 to 10 days is when the blastocyst implants and begins secreting HCG. At 2 weeks, the bilaminar disc is formed. And after 8 weeks, a lot of the basic organogenesis has occurred, so the embryo is not as susceptible to teratogens. So E is incorrect. So again, the correct answer is D, 4 to 6 weeks. And with that, we've covered everything you need to know about early fetal development.